I would like now to follow up with the first speaker after me in that session, which is Benjamin Rengstl from BioNTech, who shows a little bit the other side of application of that systems, and he will tell us about novel approaches for using mRNA for therapeutics.
Yeah, I hope you can hear me. Many thanks, Heinrich. Exactly, so hopefully I'm not disappointing you, but talk about the old school lipoplexes. But we are using them for completely different applications. So I'm talking about cellular therapies, in particular CAR T cells, and we are combining those cells with a polyplex vaccine to boost the CAR T cells in vivo. Actually, it's not the first time I'm talking here at Clinam before fully virtual, so I'm happy to be here the first time, thanks to the organizers. And this is an update on our running phase 1 trial. My disclosures, exactly. So the wall is quite big. Have to go here. So we are not doing a new pioneering combination concept. We are also addressing a new target for cell therapies, and this is Claudin-6. And as you can see, Claudin-6 is a beautiful target because it's completely absent in healthy tissue. The reason is at the bottom left, because it gets silenced during organogenesis, but it's highly expressed in a lot of cancers. At the top ranked is testicular cancer and ovarian cancer. And we have developed a Claudin-6 specific CAR. This is highly potent in preclinical models, so I have presented on it already. And we combine it with the RNA vaccine. So it's simply plain RNA, in vitro transcribed RNA that we encapsulate with liposomes. We have the lipoplexes. This is this standard BioNTech cancer vaccine technology. And once you inject it intravenously, it gets taken up by the antigen-presenting cells in the lymphatics and presented on their surface. It's the natively folded protein, so the perfect target for the CAR T cells. And our goal is depicted on the bottom right. We want to expand the CAR T cells in vivo so they are oscillating in the optimal therapeutic window. Exactly, and we are performing since more than 2 years now already a phase 1 safety tolerability trial, but with a lot of good data on efficacy. Let me guide you through this, fully aware that you're maybe not that familiar with clinical trials, but it's pretty simple. So we first test our patients for Claudin-6 expression using an immunohistochemistry kit. And once this is confirmed, we take the cells from the patients, so the leukocytes, and then in our GMP facility, those get retrovirally transduced to express then constitutively the Claudin-6 CAR. And the T cells are then infused back to the patients, and we are doing a bifurcated trial design. So it's always 3 patients per cohort, and we are starting with the dose level 1. It's 10 million CAR T cells, then going 10-fold higher to 100 million CAR T cells. And in parallel, on the second arm, we combine it with the vaccine. And in the middle, you do see the first dose escalation. This is already completed and was reported at ESMO last year with about 22 patients. And this is using a product that was produced, so a CAR T product, very manually. And because we have seen such encouraging efficacy data, we decided to change the manufacturing process. It's now more automated, or you can say fully automated. You have a better scalability, you have a better robustness, but we also saw that the product is more potent. So we have started with the dose escalation again, now even at the dose level 0. And the data that I'm presenting is actually data we have presented at ASCO this year. And there we have treated in total 19 patients with the new product. And we will have a presentation in 10 days from now at ESMO again. And there we will report on 44 patients. But it's too early to share, unfortunately. That's not my talk, it's for the lead PIs then. So regarding safety, so in the first dose escalation, we saw a very manageable safety profile of the CAR T cells. And most importantly, we didn't see that the vaccine on top is adding any toxicity. Yeah? And this could be confirmed with a new product as well, but we have not explored the relevant dose levels back then. So we, as you can see, in dose level 2, so it's the 3rd row and the 5th row, we have treated 10 patients in total, so not that many. And this is the relevant dose level, and I can show you why. If you look at the efficacy, this is like the colored bars. fully colored bars show you dose level 2 patients, and most of them are responding. Actually, there's only one patient not responding, and we are quite happy that we have now germ cell tumor patients and ovarian cancer patients. Before, we have treated way more germ cell tumor patients because simply they do not have any treatment options. So you have to keep in mind we are treating patients in the 4th or 5th treatment line in average. So they don't have any chance anymore. They have a life expectancy of 3 to 4 months. And we even have now complete responder patients or patients that go for 6 months or longer into partial remission. Then here, this is the data from the first escalation presented at ESMO. You see the other 22 patients, it was very comparable. We have a response rate of 30 to 40%, disease control rate of 60 to 70%, and this is quite striking. So keep in mind, those patients do get palliative care, so you just wait for them to die. So now I talked a lot about the CAR T cells, so let me quickly touch also on the vaccine. On all those plots, so this is patient level data. You do see in black the tumor marker. So in red, you do see the CAR T frequency measured in the blood. Yeah? So we have developed a PCR, so we can take blood from the patients and we can determine the frequency of the CAR T cells. And you see nicely here on the far left, this is our complete responder patient from the first dose escalation. So we have 2 now. Actually, we have a 3rd one last week. The CAR T cells engrafted nicely and then went into a plateau. And at the same time, the tumor marker simply dropped. And this is an ideal response. This patient was disease-free for 18 months. Unfortunately, he passed away afterwards. There was a relapse, clone 6 negative, no chance anymore. And this is how it looks like from the CT scans, just that you're seeing. So he had a very big lung metastasis, and it was fully gone. Actually, after 18 months, in the last picture, we saw a hole in the lung, so nothing was left, full metabolic response. But now coming to the vaccine, and we know that dose level 2 is therapeutically relevant. This patient here in the middle received only dose level 1, so 10 million CAR T cells. If you compare the literature, this is a homeopathic dose, actually. It's a flat dose, 10 million only. But we vaccinate. You see the green dashed lines. Those are all the vaccinations. And sometimes you do see like this little expansion, then followed by a contraction. And on the left, you do see the CT scan of this patient. He had 15 plus lung metastases, all were gone after 6 weeks. So this is the picture from the first staging. And we were super happy to see that we expanded the red curve, so the CAR T cells, to therapeutically relevant levels with the vaccine. And we were able to keep those cells alive for up to 200 days. Then the patient got redosed, and after 200 more days, he got redosed again. He was always responding. And usually, CAR T cells in solid tumors, so there's no product on the market yet, they vanish within the first 2 to 3 weeks. And we do see them for months. And on the far right, you do see another example of a patient that is benefiting from the vaccinations. So the red curve is always above the lower limit of quantification. Actually, this patient was the reason that we amended the protocol, because he's now more than 2 years on trial. And keep in mind, 3 to 4 month life expectancy. And gets continuous vaccination, and he's tolerating the vaccine pretty well. All those patients get, after 6 hours, like fevers and chills. They get a bit of paracetamol, acetaminophen, and doing great with the vaccine. So many thanks to the entire community that has developed such nanoparticle delivery methods that we can use something like that in clinical trials. And bottom line, we have here 3 examples of 13 germ cell tumor patients. And after 50 years, it's the first therapy for them, basically. There was no development from a clinical perspective. After 50 years, we now have the first product, hopefully, to bring to the market soon. Yeah. And with this, I think I can skip the key takeaway message. And of course, I would like to thank the whole study team. So there are a lot of people involved, a lot of from BioNTech, a lot of investigators, patients and families. But most of all, I want to thank you for your attention.